Audio, video, we all good? Good morning, everyone. It's my pleasure to introduce Grand Rounds this morning. On Wednesday, October 23rd, we'll start with a land acknowledgement. Ottawa is built on unceded Algonquin Anishinaabe territory. The peoples of the Algonquin Anishinaabe Nation have lived on this territory for millennia, and we honor them in this land. Their culture and presence have nurtured and continue to nurture this land. CHEO also honors all First Nations, Inuit, and Métis peoples and their valuable past, present, and future contributions to this land. So this morning, our speaker is Dr. Izabella Pena. Dr. Pena is an early career scientist at CHEO and the University of Ottawa, and her research area is focused on inborn errors of metabolism affecting the pediatric population. She started her lab here in 2023, last summer, she's in the Department of Cellular and Molecular Medicine at the University of Ottawa, and a scientist at the CHEO. And you know, Izabella's story I know quite well. She did her PhD here and then went and did a very productive postdoc at MIT, where she worked on developing CRISPR screens to investigate novel genes related to mitochondrial vitamin B6 metabolism and how that relates to disease. And you're going to hear about that today. She uses cell-based assays as well as zebrafish, which of course is the most important model you can use, but I'm not biased. And is focused on rare diseases. She has been involved with the Care for Rare consortium led by Kim Boycott. Her newly established lab applies these new technologies in cell zebrafish and mouse models of rare diseases to understand pathophysiology, search for novel biomarkers, and new therapeutic avenues. And we're really fortunate that Izabella decided to return to CHEO and Ottawa from her time at MIT in Boston. The title of her talk today is Basic Science and Translational Investigation for Vitamin B6 Responsive Disorders. Izabella.
Thanks so much for the invitation. It's very nice to be here, and I'll share some of my research. Let me just hide this thing here. What was it? Hide floating, this one. Okay, great. Let's see if this works. Oh, here we go. All right. So today we're going to talk about vitamin B6 and a number of disorders that are related to vitamin B6. We're going to talk about especially redox-independent epilepsy. Right, my thing is on. It probably doesn't shine on the screen. Huh? I guess it doesn't. So I'm just going to do like that. And
people online better.
Yeah, maybe better to do like that. I guess.
Usually, yeah.
This, they can see me, right? If I do this, perhaps. Okay, sounds good. So we're going to talk about vitamin B6. I don't know if everybody knows what is exactly vitamin B6, one of the key vitamins that we eat in the diet, but we're going to understand a little bit of the chemistry and what it does in the body. We're going to talk about redox-independent epilepsy. It's one of the main diseases related to vitamin B6. Some clinical aspects, some zebrafish disease models I made, and some insights we got from those animal models on pathophysiology. And then we'll talk about the work I did at MIT using CRISPR screens, knocking out all the genes in the genome to find what are the key genes related to vitamin B6 biology. And then some of the translational research that we're doing here at CHEO for redox-independent epilepsy. And I hope this is going to be interesting for you all. So to begin with, to talk about vitamin B6, so actually I have several vitamins, and those are the vitamins that correspond to vitamin B6. It's 6 chemical molecules. So we have pyridoxal, pyridoxine, and pyridoxamine. So they all have this pyridoxine ring and different compounds. So here's an imine group. This one is actually wrong, but this should be an aldehyde group here. And then pyridoxal phosphate, which is the phosphorylated forms of that, pyridoxine phosphate and pyridoxamine phosphate. Pyridoxal phosphate is actually the active form of vitamin B6. They can't see me? Sorry. They can't hear me. Okay. So pyridoxal phosphate is actually the active form of vitamin B6. So that's the one that acts as a cofactor for a number of enzymes in our body. And we usually eat those from the diet, so we cannot synthesize those vitamins. So we have to obtain them from the diet, from plant-based or meat-based diets, we can get those vitamins. And they get absorbed in the intestine. They go to the liver, where the liver synthesizes PLP from either the unphosphorylated forms, for example, and then it binds to albumin, and that's where it gets into the circulation. And we actually get PLP to the tissues in the form of PLP bound to albumin. And then we excrete vitamin B6 in the form of 4-PA, which is 4-pyridoxic acid, and that's the main excreted compound in the urine. So to begin with, those vitamins have to enter the cells, and we cannot enter the cell in the PLP form that is bound to albumin. So we have phosphatases in the membrane. So one of them is called TNSALP here. And those phosphatases remove the phosphate group, and then it enters the cell. And then we have a pyridoxal kinase that adds the phosphate again, phosphorylate those compounds, and that's how we trap the vitamin in the cell, because the phosphorylated compounds cannot exit the cell. It's analogous to how glucose enters the cell via hexokinase. We trap them in the phosphorylated form. And then PLP acts in the PLP-dependent enzymes. It binds to this other protein called PROSC that we don't know very much what it does. And it goes to the organelles, for example, mitochondria, where there's another set of PLP-dependent enzymes. And here's, I put a few stars of some of the diseases that are caused when we have, mutations on genes related to the vitamin B6 homeostasis pathway, and they all cause epilepsy. And we're going to talk about that. And so what PLP does in the cell is many various functions. First, we have about 60 enzymes in our human genome that depend on PLP for their activity. And they depend on PLP as it binds to the active site. It forms a shift base with a lysine residue in the active site. And it catalyzes several reactions from transaminations, racemization, cyclin cleavage, decarboxylation, a number of reactions that are necessary to have PLP in the active site to bind. And those enzymes, you can name your favorite pathway. I don't know, you like muscle, glycogen phosphorylase, for example, is a PLP-dependent enzyme, critical for glucose metabolism, right? Glycogen metabolism. Or neurotransmitter biosynthesis. There's a few enzymes that depend on PLP for several catalytic activities. And we also have PLP-dependent enzymes in several compartments in the cell. So all the ones that I'm showing there in red are, for example, localized in the mitochondria. So a third of all the enzymes actually are mitochondrial. So showing the importance of this vitamin for mitochondrial metabolism. And as I mentioned, a number of diseases are related to vitamin dysfunction. So actually the vitamin Bs are very important, especially for mitochondrial metabolism. So for cofactor metabolism, for several functions in the mitochondria. We hear that mitochondria is the powerhouse of the cell, but none of the powerhouse functions, the bioenergetic functions, can be achieved without vitamins. So the vitamins are actually catalysts that, for example, allow the electron transport chain to function, like heme, iron sulfur clusters, all those metabolites, they're important for function of the mitochondria. And we have a lot of diseases related to primary mitochondrial disorders that are related to vitamin dysfunction. So for vitamin B6 epilepsies that I mentioned that is one of the focus of my research, we have about 4 genes that are known to cause those types of diseases. And the majority of the diseases related to vitamin dysfunction in the mitochondria lead to neurological implications from age 0 months to 3 years old. So the vitamin B6 epilepsies, or B6DEs, they're rare epilepsy disorders. I'm going to talk about one of them in particular, mutations on ALD871, that gene that I'm showing there with a red cross. So that's a gene on lysine degradation. So if you have this gene missing in the genome, lysine cannot be degraded efficiently. And, lysine is an abundant amino acid that we get from the diet, and it gets degraded, as we speak. But in patients with mutation on this gene, lysine gets completely degraded. So this compound here, ASA or P6C, gets accumulated because it cannot be converted by ALD871 into an acid that is not toxic at all. But this compound is very toxic. So when you don't have this enzyme functioning, this compound accumulates, and then it causes a very bad thing, which is the complex with PLP, vitamin B6, forms a chemical complex that just eliminates the active form of vitamin B6 and causes a vitamin B6 deficiency in those patients. And the result of that is that patients have neonatal recurrent seizures. So they start right after birth, a few hours after birth, sometimes a few days, like 2 or 3 days after birth. Those are seizures that are resistant to any anti-epileptic drugs. So it's a very recurrent and resistant to any medication. But those seizures are uniquely responsive to vitamin B6. It's kind of a magical thing. You inject, do IV injection of vitamin B6, I think it's 100 milligrams, and it immediately ceases the seizures in about 10 or 15 minutes. And that's very remarkable. And that's what happens as well with mutations in all the other 4 genes. So it's a pretty much similar presentation, early neonatal seizures that get alleviated by vitamin B6 injections. And then the patients require vitamin B6 treatment every day for the rest of their lives. And that's the only way that the patients can survive, but they do survive. There's some other problems too. So those seizures are mostly alleviated with vitamin B6 treatment. They still have neurodevelopmental problems. There's some cognitive disabilities, some intellectual disabilities. Some patients have autism. There's some other, neurodevelopmental problems that are not addressed with vitamin B6 treatment, likely due to the accumulation of those metabolites, which are likely. All right, so to investigate how this disease occurs and how vitamin B6 metabolism is defective in this disease, we decided to create zebrafish models to investigate epilepsy. And then you may ask me, like, what zebrafish in epilepsy? How do you look into epilepsy in zebrafish? Like, fish can seize and have, convulsions? Yes, they can. And not only they can, because they have very similar brains to our brains. And we have about 70 to 80% genetic identity with the fish. They're also very small. So this is how tiny zebrafish larvae are. And you can actually put them in 96-well plates. Like you can put one fish per well in a 96-well plate and actually treat them and do several types of assays with those fish. So in here, I have an example. We put, each well has one fish larvae. So we're talking about pediatric diseases, right? So the fish are gonna have disease when they're larvae or embryos, right? So those are the tiny larvae, they're about like 1 millimeter long. And you can put them in wells. And here I'm adding a drug called PTZ, pentylenetetrazole. That's a GABA receptor antagonist. And what it does, it induces seizures, right? And seizures in zebrafish manifest as very fast swimming movements with very fast, jerky convulsions that you can see in zebrafish. I don't know if you guys can see here because the thing is not the best, but you see the fish swimming very fast here. Let's see if there's this one. I don't know, it's not the best here. Can you see it? Okay. So that's
we can see, but we can't hear.
The plate, and we have an infrared, and then we use a computer vision software to actually design traces of the movement. So in red, we have fish that the movement that was traced by the software in very high speed movement, and the green is just like medium normal speed. And you can see that the fish that receive the drug, pentylenetetrazole, especially high dose, they're moving very fast. And we can convert the traces into numbers and plot this as a bar graph or like a dot plot. And you can see that the higher concentration of PGV induces a higher distance move in this very high speed. So we can use these types of assays to investigate zebrafish epilepsy. So using CRISPR, which is this technique now that everybody talks about, we can actually introduce mutations in the gene of interest to deplete the gene to create like a deletion or loss of function mutation in the fish. So we did that and we created a mutant line that has 5 nucleotide insertion mutation that leads to complete ablation of the protein. So they have an early stop column and they don't produce the protein at all. ALD871. And then the result of that is that they do have the biochemical phenotypes. So they do accumulate the toxic compounds that are supposed to be accumulated when you don't have ALD871, which is the same that patients have. And then they also have the spontaneous seizures or epilepsy. So, so if you can see here, the ALD871 fish, they're having those very fast movements and they're having the seizure behavior that we expect to see when a zebrafish is having epilepsy. But the wild type never do that. And they start having that around 10 days post-fertilization in the larval stage when they're still little babies. So we can actually trace that using the software, as I mentioned. You can see a lot of red lines here, which shows that they're having very fast movements and very hyperactive behaviors. And we can actually, oops, oh, you got it. It's because this doesn't work. And then the other thing we can do with the zebrafish to study epilepsy is to do electrophysiology. So, you can do kind of an EEG in the zebrafish brain. So you get the little zebrafish and we get an electrode and we poke their brain to do put a field recording of the brain so we can measure the spikes of the electrical activity, showing when the neurons are all spiking together. That's basically when the seizure happens, right? Like have very strong electric signals, and we can detect that by using electrophysiology. So we do this and we can only see those massive spikes of electric activity in the neurons in the mutant fish, never in the wild type. And then we can do also metabolomics. So the question is, when you're talking about vitamin B6 biology, we're thinking about metabolism, right? So this vitamin is very important for several reactions, as I mentioned, about 60 enzymes in the human genome. Well, which of those enzymes is related to epilepsy, right? And when we do metabolomics of their little zebrafish heads, what we can see is that the mutant fish have very low levels of GABA, which is a very important neurotransmitter. It's our main inhibitory neurotransmitter, and it's very important, in the seizure context. So low drops in GABA levels are known to induce epilepsy, and we all know that based on how the brain works. And this makes a lot of sense if we think about how GABA is synthesized in the brain. Oh my gosh, why is this not going? Sorry. So GABA is synthesized in the brain from glutamate, another amino acid, by this enzyme, glutamate decarboxylate, or GAD, which is a PLP-dependent enzyme. So this enzyme needs PLP to make GABA. And we see very low levels of GABA in the fish, in the mutant fish. But when we treat them with pyridoxine, we actually can restore the GABA levels. So that may be one of the mechanisms why there's epilepsy in this disorder. And it's very striking to see that the patients, when they receive pyridoxine, they actually survive, right? We can have a complete cessation of seizures. In many patients, there's still some residual seizures, but most patients will have complete cessation of seizures, and the pyridoxine treatments every day enables life. The fish is the same. So here are the fish, the mutant fish, they die around 14 days post-fertilization. But if we treat them with PLP, which is the active form of pyridoxine of vitamin B6, we can actually increase the lifespan. So there's a dose-dependent increase in lifespan. PLP is not very soluble in water, so that's the highest concentration we can do. But pyridoxine is very soluble, so we can do higher concentration. And then if we do pyridoxine, for example, at 1 millimolar or 5 millimolar, we have complete rescue of survival to fish to adulthood. I could actually get those fish to reach adulthood, breed, have little babies, so they actually have a normal life. I don't know if they're having, the cognitive functions that the fish have. We don't know how to measure that, but we'll figure it out. But yeah, so it seems to completely restore the fish in the pyridoxine treatment. And it also seems to restore the epileptic behavior in fish. So this is how the electrophysiology looks like in the untreated fish, and that's when we look into the fish that are treated with vitamin B6. So you can see that the event duration, which is the duration of the long spikes, they drop massively, and the number of events also drop massively. So it also shows the rescue of the electrographic component of the seizure phenotype. I'm going to talk briefly about another cause of B6 epilepsy, which is caused by this gene called PROSC. And that was a story that started here at CHEO. So there was a neonate with the medication-resistant seizure that was admitted here. And this patient had the mitochondrial encephalopathy-like disorder, had like very strong lactate peak, and it was not thriving well. There was some sort of movement disorder too, and it was a very confusing picture, because if you look into the other epileptic patients from PDE, for example, there's no much of a movement disorder or even this mitochondrial encephalopathy. It's not very common. But then with care for rare, we found mutations on this gene called PROSC. And that was an orphan gene at the time. We didn't know what it does. And then we realized that this is a novel form of pyridoxine-dependent epilepsy. So reach out. This was a project that was led by Devon Johnston. So he was a PhD student. And I was co-leading with Claire van Karnebeek, who is a clinician now in the Netherlands. She was in Vancouver at the time. And she actually found another few patients, and we combined all the 12 patients we described in this manuscript, showing these new mutations on PLP-HP, also called PROSC, as a new form of pyridoxine-dependent epilepsy. So the was very confusing, because we had from completely normal MRI to very abnormal pictures, and the mutations were also varied there in several places in the protein. So it was really hard to find out what is the genotype-phenotype correlation. The other thing we could find is that the truncation mutations were very severe, and the mutations around the PLP binding site seem to be also very severe. So, but some of the mutations seem to still have some residual activity. The thing is that we don't even know if this is an enzyme or anything. We don't know what this protein does exactly. We know it binds PLP very strongly, but we don't know what it does. We think it's involved in homeostasis of PLP, potentially like a chaperone of PLP to the other enzymes, like to deliver PLP to the other enzymes, something like that. And we made a few laboratory models of this disease. One of them was using yeast. And with yeast, we saw that yeast can grow. This is the PROSC homolog in yeast. And it can grow very well in glucose, but it cannot grow in carbon sources that require mitochondrial metabolism. And we also saw in human cells that PLPHP, which is PROSC, is also localized in the mitochondria. So this may be related to the mitochondrial encephalopathy picture that we saw in the patients. And then we created the zebrafish model as well that had very similar phenotypes as we saw for the OD871. They actually had, the seizure behaviors. They died very early, similar to the OD871 fish. The hydrographic component of the seizure phenotype was very clear. And when we did metabolomics of that, we also saw this very strong depletion of GABA levels in their brains. So it's very similar. But in addition to the GABA levels, something we didn't see in the OD871 fish was a striking increase in the substrates of this enzyme here, AADC, which is aromatic amino acid decarboxylase. And that's the main enzyme in the brain for the synthesis of dopamine, for example, which could be related to the movement disorder that we see. So it's very likely that the patients have not only a deficiency in GABA synthesis, but also in dopamine synthesis. And that's probably why we had this movement disorder picture. And that's a PLP-dependent enzyme. I don't know, I went to the last slide, I don't know why. Sorry, how do I do this? I'm sorry for that. Okay, I'm getting it. I'm sorry. I was gonna get to the most fun part now. Okay, so I showed you two examples of how, treatment with vitamin B6 can really change the lives of the patients. So most of those patients, when they are treated, they actually have complete recovery of their epileptic phenotypes, and they can, have a pretty normal life despite all the, the other cognitive disabilities they may have. And it's the same that we see with the fish for both models that I showed, the OD871 and also PROSC. Treatment with pyridoxine really restores their life and enables life. And that was a transformative effect. And the question that I had here is that, are there other genetic disorders that are caused by, genes that are important in the vitamin B6 biology that could, I don't know how to do this, here we go, that could potentially benefit from this treatment, which is such a cheap and simple intervention. So as I mentioned, PLP is an essential cofactor for life. I mentioned being a cofactor for neurotransmitter biosynthesis, such as GABA and dopamine, which are relevant neurotransmitters for the diseases that I just talked about. But this is also a very important cofactor for several other pathways, including mitochondrial pathways such as the heme biosynthesis pathway and isotope cluster biogenesis. There's two very key enzymes in both pathways that require PLP. And those are very important for anemias, such as roblastic anemias, which are caused by deficiencies in those two enzymes. And then a way to investigate in an unbiased manner which all the genes are essentially in vitamin B6 stress is using CRISPR screens. And I mentioned about CRISPR that we used in the zebrafish to knock out one gene, right? But there are ways nowadays to knock out all the genes in the genome at once. And that's using pooled CRISPR screen libraries. So basically we have a library with sgRNAs, which guide the Cas9 to go cut the gene, the target gene in the genome. But we have a pool of them and they're all barcoded and you can work with them all together in the lab. So what we do basically is the other cell lines, such as K562, that's a very important cell line. It's an enriched leukemia cell line. It's being used widely for CRISPR technologies. And we infect those cells in a way to get one cell receiving one guide RNA only. So each cell is going to be knocked out for one gene in a population. So this is a very big bucket of cells. Imagine like 1 billion of cells in a bucket. Each one is a knockout for one gene. And then we grow those cells either in a minus B6 condition or plus B6 condition, like a superphysiological condition. And then what we see is that we ask in the end is which cells survive and which cells don't survive. So the cells that survive are probably cells, when you knock out the gene, it doesn't affect that phenotype, right? It doesn't affect their growth in minus B6 or plus B6. But if, let's say you knock out this blue gene here, and the cell did not survive in the minus B6 condition, probably that gene is important for surviving minus B6, right? So for example, this could be pyridoxal kinase, or it could be the transporter for vitamin B6 to get into the cell. So, like when we think about how we discover the diseases that are responsive for vitamin B6, like how do we find out that PROSC deficiency is responsive to B6? Isn't based off trial and error, right? Patient gets to the hospital, we don't know what gene mutation he has, and then you try something, and then it has a responsive, and then you go and sequence and find out the gene. So that's kind of forward genetics, right? You find the phenotype first, and then you find the gene cause. So here we're doing kind of a reverse genetics. we knock out the gene, and then we find the phenotype that it gets rescued or not. And maybe that gene is going to be responsive to vitamin B6. And that may be caused by, that gene mutation may cause a disease that happens in clinics that could be responsive to vitamin B6, right? So that's the rationale why I'm doing this. So by doing this, I knocked out all the genes in the genome in a big bucket of cells, and then we sequenced them, and we found out which genes are important in vitamin B6 deficiency, which is the gene score in minus B6, this line here. And then we have the gene scores in plus B6, which are the genes that the cells are growing in high concentrations of vitamin B6. And then we found out that, for example, PDXK, PNPO, and PROS, which are genes known to be in the vitamin B6 pathway, they're actually very important for vitamin B6 biology because they have negative gene scores in minus B6 condition. And then we found out this gene here, SOC25A38, that seems to be very important. They have a very negative gene score in the minus B6 condition. And we're going to study that one a little bit more. But to begin with, we should see that most of those genes that have high gene scores, they're actually mitochondrial genes. So showing how important vitamin B6 biology is for mitochondria. And so to replicate this, we did a small library screen. So we just replicated the whole result. And we found out basically the hits that we found in the big screen, they found again in this small targeted library. And then we're going to investigate now SOC25A38, which is a gene deficient in a disease called congenital sideroblastic anemia. And that disease, in that disease, there are patients that come to the hospital and they have very severe anemia. They need blood transfusions like very frequently, probably like every 3 weeks or so. And they do have a striking accumulation of iron in the mitochondria. They cannot synthesize heme very well. And the synthesis of heme, as I mentioned, depends on PLP. And this gene was supposed to be the glycine transporter that brings glycine to the mitochondria for this first committed step of heme biosynthesis, which is basically combining glycine with succinyl-CoA to form this compound called aminolevulinic acid. And that enzyme called ALA-S is a PLP-dependent enzyme. So because this is such an important step in the heme biosynthesis, I was asking myself, what if this protein is also regulating the import of PLP in the mitochondria? Not only glycine, but it's also doing some other functions that we didn't know yet. Otherwise, why would it be so important in minus B6 biology, right? In minus B6 screen. So then we knocked out, now using the CRISPR to just knock out one gene. So we knocked out this protein, and then we created knockout cell lines. And those cell lines indeed do not grow with B6 depleted media. So you can see the yellow bars. They still grow in the high B6 media, but not in the B6 depleted. And then to ask what is the status of vitamin B6 in the mitochondria of those cells, we decided to investigate using this very cool technique called the mitotag, the mitochondrial IP. So we get cells that have a little tag in the mitochondria. So we create a genetically encoded mitochondria. And then those cells have this tag that we can bind to an antibody bound to magnetic beads. And then we can pull down those mitochondria, the entire organelle. You know, when we pull down a protein, now we're here pulling down the entire organelle. Then we can wash that and open just the mitochondria and look into the mass spectrometry to see what is inside the mitochondria. And by doing this, we found that the mitochondria from wild-type cells have PLP, pyridoxal phosphate, but the mitochondria from SOC25A38 cells don't have PLP in the mitochondria. So it's very low, very close to the background. And so that showed us that maybe this protein is playing a role on the accumulation of mitochondrial PLP. And we saw that this low levels of mitochondrial PLP can be rescued if you add back SOC25A38, if you add back HAM25, which is the yeast ortholog of SOC25A38, but not if you add another kind of protein, for example. And we also see mitochondrial PMP following the same pattern. So PMP is formed in the active site of transaminases in the mitochondria. So when you get an amine group from a metabolite, it goes to PLP, forms PMP, and then it passes the amine group to the other substrate. And so we got a few congenital sideroblastic anemia point mutants, and we created those point mutants mostly in residues that affect the activity of the transporter function. And we saw that none of those point mutants can rescue the low levels of mitochondrial PLP or PMP, only the wild type form can rescue the low levels of PLP. And then lastly, we did some stabilized isotope tracing to ask the question if another protein in the vitamin B6 biology pathway is missing, is messed up, or is it just the import in the mitochondria that's messed up? So we use the labeled pyridoxine. So instead of using a pyridoxine that has hydrogens, it has deuterium. Deuterium is heavier, so it's 1 unit of mass heavier. So this is a pyridoxine that is 3 units of mass heavier. So we call it M+3. And then pyridoxine gets imported into the cell, gets phosphorylated by pyridoxal kinase, forming PNP, the heavy form. And then it gets converted into PLP, the heavy form of PLP, and then gets imported into the mitochondria. So we asked if those steps are messed up, or this last step is messed up. And we saw that in the whole cell, which is the input of our IP, we see the M+3 pyridoxal phosphate, showing that this step exists. So this is functional, but we do not see the M+3 step, pyridoxal phosphate, into the mitochondria. So showing that this step is not functional. So likely there's a problem in the accumulation of mitochondria in those knockout cells. Could it be that it's associated 538 is the transporter for mitochondrial PLP, or maybe it's regulating the transport of mitochondrial PLP, or even regulating the accumulation itself, like regulating degradation or something like that. And I'm not going to talk about that. So this is something that we found using CRISPR screens. We found a novel regulator of mitochondrial PLP, homeostasis in mammalian cells. This is the most essential gene in vitamin B6 stress in those cells, GIF6-2s. The knockout leads to severe growth defect, and it's rescuable, and it's condition independent, leads to low mitochondrial PLP levels. And we believe that this is a very important protein for vitamin B6 biology that was not known before. And that's important because, as I mentioned, associated 538 loss of function leads to congenital stereoblastic anemia. So this is how the erythroblasts of those patients look like. They have those rings of iron around the cells, and those are actually iron deposits in the mitochondria that we can see here. But what if the problem here is that there's a low levels of vitamin B6 in the mitochondria, leading to this whole scenario, in addition to an effect on glycine import that was shown before in this disease. So now we're very excited about this to try to take this knowledge further, create zebrafish models, create other mouse models, and try to see if rescue with vitamin B6 is another therapeutic option for this disease, maybe to even improve the timing between transfusions or something like that. And so that's what I'm doing in my lab. So we're studying vitamin and cofactor biology. So as I mentioned, vitamin B6 is the main one of them, but I'm also interested in the other vitamin Bs. And we're creating several new screens that we're going to do to investigate mitochondrial homeostasis. And we're also developing cells and animal models, so mostly in zebrafish, to create new disease models for those, to investigate that, and also create new tools such as the MitoTag zebrafish to investigate mitochondria. So for associated 538, we're creating this zebrafish model. It's actually with Jason here, he's helping us a lot with that, to try to create a zebrafish model to investigate this hypothesis that there is a deficiency in mitochondrial PLP, and also to test therapies to rescue the anemia phenotypes. We're also doing some other screens in my lab. So I did a few other screens to investigate more aspects of vitamin B6 biology, and also to investigate other vitamin Bs, which we have a lot to know. And then some on the translational side, we're trying to investigate some new approaches for newborn screening. So as I mentioned, for PDE, for pyridoxine-dependent epilepsy, those are diseases that start in the neonatal stage, and the patients have, a lot of patients go through a very long diagnostic odyssey. They have a lot of seizures until they're actually treated with vitamin B6. And as you know, like prolonged seizures can have very detrimental impact in the brain. So the earlier we can diagnose and the earlier we can intervene, the better the outcomes for the patients. So there's a few biomarkers that seem to be very stable and detectable in the newborns and the dry blood spots. And we're now doing some pilot studies to see if we can implement this as a new screening approach for the newborn screening here in Ontario. And then we did a few drug screens as well for ALD71 deficiency to see if we can improve not only the vitamin B6 deficiency, but the accumulation of the toxic metabolites that seem to be neurotoxic as well. And we found a drug that seems to be very safe called fenofibrate. And this drug seems to be able to rescue the fibroblasts, patient fibroblasts, and also rescue the toxic metabolite accumulation. So that's a drug that we're going to be trying now in vivo, and then hopefully move to clinical trials in the near future. And we're also trying to inhibit the upstream step in the pathway. So this is from PD as well. You remember this is all about lysine degradation, and that's where the mutation is, and we accumulate those toxic metabolites. Then there's an enzyme upstream called AASS, and that's an enzyme, when mutated, cause just a benign variation of metabolism. So people accumulate lysine when they have mutations on AASS, but they're just fine. Nothing happens. So we're just thinking that maybe we can inhibit this enzyme as a new therapeutic approach to avoid the accumulation of the toxic metabolites in this disease. And maybe that would help, treat the other issues that the patients have. So I solved the structure of this enzyme, and we're trying to do some, drug screenings. That's in collaboration with Newcastle and Oxford. Yeah, so this is more or less what I wanted to share about vitamin B6 biology. And oh my gosh, and lots of people think, especially my little lab that is just starting, and everybody at CHEO that I just absolutely love working here. And newborn screening, we have very good partnership now, and we hope to, maybe bring this screening technology for redox-independent epilepsy, and also my former lab at MIT. That's where I developed all the CRISPR screens that I showed here. Yeah, that's it. Thank you so much.
And correct me that you misspoke that it was your first postdoc that you did here at
Yes,
CHEO,
I did.
Not your PhD, you're a PhD in Brazil,
Yes.
and it's just amazing to see how much work you've done in the short time you've been
Thank
here
you.
as a PI, and I think you made the public topic very accessible. And when our mothers were telling us to take our vitamins,
That's why.
that was good advice. Questions, Izabella? There may be questions online.
Okay. No, there's no questions online, I think. Yes, Rob. Thank you.
So just so we can
Thank you. Yeah.
I'm just thinking about that.
Yeah. No, it's very unlikely that, for those enzymes, I don't think any of the enzymes could remove those deuteriums. So I think it's very unlikely. Yeah, they're pretty stable. In those hydrogens are pretty stable, actually. Yeah, thanks.
Yeah, congratulations.
Thank you.
For these findings, about the kids that have responses, they also have.
No, they don't. And that's a very, very good question, because so SL638 seems to be a very erythroid specific gene. So you don't have this gene expressed in neurons, for example. So I do think that there's another system that is redundant in the neurons and in the brain that does the same function as SL638. We actually may have found this other gene in a screen that I did in the lab, and we're now studying that one. Yeah. Or if it's just specific to anemia, right? Yeah. With the erythroid line. Yeah, so I think that in our zebrafish, it's going to be really specific to the erythroid line, because this gene is very erythroid specific. But I think once we knock out the other gene that I think does the same function, but in the brain, that one is going to have seizures. I think so. Thank you. Go ahead. Yeah, so one of the things we want to do, let's say, once we get the zebrafish models, we can actually do like seahorse with the whole zebrafish and see how they respire and, you know, see all the aspects of mitochondrial metabolism. So yeah, I'm gonna work a lot with Dr. Harper. So yeah.
Divorce and divorce.
Yeah.
It's like a whole, it's like under the
All the aquatic stuff.
I think so it was really great. Really good. Presenting the.
Thank you.
So my question is around.
Mhm. Yeah.
From the.
Yeah. Yeah. Safe and cheap. Yeah. 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 Yeah, so you know, as you know, like I'm not a clinician, I'm just a basic scientist, but I'm partnering with clinicians like Jason and Alex, and I think this partnership is what's going to make my science go to the translational side, right? Like, for example, with Alex, and he's now trying to write a clinical trial application for the phenofibrate that we found in the lab, and hopefully with Jason, once we get the zebrafish, we're going to do some tests and see if there's any rescue with the vitamin B6 and the zebrafish models, and then maybe we can plan a clinical trial. So Jason has done a clinical trial in the past with the.
So just a comment. When you use vitamins or nutraceuticals, the ability to actually go and take and fix some of the we did a transient model of and found that we could rescue the phenotype with folic acid and glycine. So we went back to patients when I was in Halifax, and we actually did a clinical trial on those patients, providing glycine and folic acid at doses and sort of whatever useful. There are lots of things glycine is actually used for alcohol, and also used those all approved dosing. See a rescue. So maybe maybe one of the most exciting about this with more.
That's true. So we'll see how it goes. And for the phenofibrate one as well, phenofibrate is also an FDA approved drug. So we use the approved drug libraries and it screens in the cells. And that's another drug that's probably going to be fine to go in the trials because it's safe, it's approved, it's been used in clinics for many years. So it's like a repurposing approach. So yeah. So see, so the hope is that with this type of research, we can do a bit of the reverse genetics and find, genes that may be responsive to treatments before even knowing that the patient can be responsive to treatment, right? Like doing this in the lab and then move that to the clinics. That'll be the idea. Yeah. With supplements, which is simple, cheap, safe, right? Yeah. Right.
Any other
Questions?
Let me see if there's anything.
Alex, do you have a question? Yeah, feel free to ask, guys. Don't be shy. I hope it's not because it was confusing.
No, that was great. Well, thanks everyone. Thank you, Izabella, for that information. And the evaluation. the chat or discuss it online. Have a great day.
Thank you. Should I just end here?
Yeah.
Okay.